to study time is actually, I'm not good at waking up early in the morning since when I was a high school student. So I study like, yes. And so today I showed that some of the techniques of the physics can be used in biology, and prediction was verified. So please imagine you are in a cave, the 300 steps from outside. The light intensity is constant and temperature is constant. Can you wake up tomorrow or day after tomorrow? Can you wake up tomorrow? I'm not sure. Yeah. I think the many of you can wake up tomorrow and day after tomorrow without the information of time. Why? Because we have a daily rhythms in our body, and it is called the circadian rhythms. And so what are the components of the daily rhythms or the circadian rhythms? Actually, genes and proteins in the biochemical reactions. Let me explain it. So from gene, you see, you know, protein, you eat lunch and including protein, and within a cell, you make proteins. So yeah, so from RNA, this is a precursor of the protein. The protein is made, and proteins can be, technically modified by addition of phosphate in Sanka in Japanese, and it downregulate the gene expression. This can cause the autonomous oscillations in gene activity with a period around 24 hours. This is a mechanism. Did you understand? Yes, yeah. So let me explain again. And so suppose abundance of protein is high, downregulation works. Then gene activity becomes lower and lower. Then abundance of the protein becomes lower. Then now the downregulation can be weaker. So gene activity becomes higher and higher, then protein becomes larger and downregulation becomes stronger. This is the mechanism, right? This was found first in the fly. And later it was found also in plant and also mammal. And many studies have been going on, and open problems first is the about the diversity of the sleep wake timing. If you are normal, you may sleep around the 11 p.m. or the 11 p.m. or 12 a.m. in the evening, and wake up around 7 a.m. or 6 a.m. in the morning. And if you are advanced type, it doesn't mean there is a superior, but you sleep around 8 p.m. in the night, and you wake up around 3 a.m. or 4 a.m. in the morning. There are such a family. So if you are delayed type, delayed, and if you are non-24-hour type, your sleep schedule is shifted every day, even in a 24-hour modern society. Around not all, but half of the blind people is like this type, but still they can be happy with some clock and also the temperature change. If you are narcolepsy type, your sleep duration is more fragmented, 2 hours or 1 hour. Sudden sleep can occur for the people with narcolepsy. And the challenge is to understand these various sleep types. Genes play many things, but gene doesn't determine everything. And also the habit, the coffee in the night, or the alcohol, and also the aging matters. So to understand this, the theory can play a role. Other challenge in this field, I mainly talk about this today. This is about the stability of the period of the daily rhythms. Many things in our body, many phenomena in our body becomes faster at high temperature. Suppose if you put milk outside in summer, it will be easily rotten. It is because the enzymatic reactions becomes faster at high temperature. So in general, and as the temperature increasing 10 degrees, gives the reaction speed twice or 3 times increase in the reaction speed increase. And on the contrary, the period of the daily rhythms is almost unchanged against the change of temperature. Then it is a mystery. To understand this, we introduce an index like this. Nowadays, in biology, we can have the high time resolution data engine activity. So we try to make use of it. This index, you don't have to remember this, but let me explain. So this is just indicate the degree of the distortion from sinusoidal wave. Just like this. If this far from this sinusoidal wave, the index becomes larger. So yeah, as all of you know, you can have coffee, but so any time series can be expressed by a summation of the trigonometric function, right? So that like this ends also for the protein abundance ends. AJ is the Fourier coefficient, and if it is just a one sinusoidal wave, it is 1/1. If the number of components is larger than 1, this ns is always larger than 1. Such an index. And from the model for this one, we can actually understand how waveform can be changed. And so I think X1 is abundance of the RNA and X2 is the protein. And P1 is the synthesis of the protein and P2 is the rate of the modification. And the K1, K2, K3 is the decay constant is assumed to be linear for the simplest, right? And because this equation is very simple, actually this is the simplest equation for the organisms. From this we can, so f of x3 can be anything as long as the oscillations can come. And from this we can obtain a period. This is exact formula, but a bit technical, but just trust that this K1, K2, or K2, K1, K2 plus K2, K3 plus K3, K1, but K1, K2, K3 is just decay rate. Because these appear in denominator, as the reaction speed becomes faster, period tend to be shorter, right? So actually to incorporate the temperature effect, so the reactions is expressed as like this, the following Arrhenius law. Okay, as the temperature becomes higher, the reaction speed becomes faster. Now, what is required to obtain the stable period? The requirement is actually the waveform distortion. Waveform should be more distorted at higher temperature. That is what this equation says. Why? Because at high temperature, this part is larger. To maintain this period, this part should be larger to cancel out the effect. Okay, so our prediction is the waveform should be more distorted at high temperature, and if you measure the gene activity, and actually it's consistent with experimental data in fly and also mouse, and we published this. And to show this results, we actually use the renormalization group method. When I give this kind of talk to you, some of the physicists and mathematicians often ask how did you actually use the RG equations? So let me explain for the next 5 minutes or so. So our RG, so as you know, the RG method is the the RG method is originally proposed in QFT. What is QFT? The quantum field theory. Why I'm laughing behind. We use a part of the mathematical techniques in RG method, not the whole method. So and also the framework extends to the ordinary differential equation and partial differential equations, and our collaborator Kurosawa systematized the method. And actually, this sum the perturbative solution yielding an approximately valid solution for the global timescale. And in this approach, arbitrary time and integration constants responds to renormalization point and renormalization constants in QFT. this actually, this sentence is written by Hatsuha-san in Japanese and translated by me. And so what we did actually is suppose f of x3 is like this form with the nonlinearity parameter n. The condition for the oscillation is actually large n. We need nonlinearity to explain oscillations. And the condition is like this, it is less than 0. And perturbative, when we do the perturbative expansion, these are just a conventional approach. Perturbation expansion with respect to n and the state variable. N is equal to the n0 at the critical point plus epsilon and x3 is x0 at the steady state plus epsilon u1 plus epsilon squared u2 plus so. And this can be solved. And actually the form is like the U2 is form of the U2 is like the T0 is the initial condition, and like this, H2, these are the parameters. These can be determined from the parameters explicitly, and A is amplitude of T0 is initial condition, and T minus T0 cosine, this one, T plus theta, T surface. This can be problematic in general. Why? Because when T is close to T0, this approximation can be very close to the exact solution. But it far from this value, it deviates. To solve this problem, we use the RG equations. The RG equations, because of this, the integral constant A theta 0 and T theta T0 can be determined so that the exact solution is satisfied for any initial condition. And this is what this equation tells. The derivative of the x3 with respect to T0 is equal to the x3, the partial derivative of the T0 plus the amplitude and the phase. This needs to be 0. This is the RG equation. And actually, this gives a constraint for the parameters that can be satisfied for any choice of initial conditions. Okay. So by using this, I skipped the detailed result, but by using this, we can obtain the time evolution explicitly. And also, we can show by using other model, this relation hold. What is this? This is NS actually, the relation between the period and NS. So for the case that what is this? Lotka-Volterra model in ecology is lion and rabbit. Lion is rabbit, and when lion is rabbit more and more, the abundance of the rabbit decreases, and then lion decreases, and oscillations occur. And so if the predation speed increases, actually the period should be shorter and shorter. If certainly talking about lion, but if the predation speed becomes faster and still the period is maintained, we should observe the waveform distortion. That is what this equation is. Because these relations can be seen in these different types of the model, we think that we thought that this can be experimental observed, and actually the prediction was very well experiment. So by use, so I show that based on the molecular biology mechanism, we make the model and prediction was very well experimental. And also this similar approach can be also powerful also for the problem of the cell division to understand actually impairment of the cell division is actually cancer. And also the similar approach can be fine, but still this way of the approach is recently challenged. This is an open problem. So implicitly we assume that the protein can bind other protein in a test tube. This is, actually we see the reaction, we assume that the reactions that occur as if reactions in cell should occur like the protein in a test tube. And actually, protein exists within a cell in a more crowded environment. It's like you know Yamamote line? Yamamote line at 8 a.m. weekday. So crowded. So maybe you may feel the difficulty to go to the door at the Shinjuku station. Yeah. So but some the situation of the proteins like this within a cell. So I think it should be fair to say that we don't really understand how should we describe biochemical reactions. And I believe that this can be solved by measurements and mathematics and physics. Yeah. And I hope that some of the challenges in this. So let me summarize. Today I want to show that some of the techniques in physics can be used in the important biological problem. And eventually this can be done because of the two reasons. The first is our the collaborator, the Shingo Ibo, and also others are very smart. And the secondary, iTHEMS is iTHEMS. Yeah, thank you.
Thank you very much. Yeah, it's a very nice collaboration among the biologists and physicists. Okay, so first questions from non-experts. Non-expert means what does it mean? Not physicist or not maybe not non-physicist, non-biologist. Yeah, but then yeah. Okay, so accept questions from anybody.
Yeah.
Yeah. Okay. Please. Yes.
Yeah, the solution you get by the origin method is in exact.
No, it's approximate solution. But
What controls the accuracy of it?
it's
I mean, conservation theory is easy, right?
so yes, yeah.
In the or when you apply the origin, it becomes.
So it actually, we don't know actually what controls the accuracy, and we thought that epsilon should determine the accuracy, but somehow if the epsilon is very large, still our approximate solutions can be very close to the solution given in numerical simulations. And so the Kurosawa actually systematized, showed in the recent book about the accuracy. Yeah, the answer is we don't. I thought that epsilon should be done, but actually it is okay for the global landscape.
Oh, so fast, yeah.
Any criticism can be fine. Yeah.
So I didn't quite understand that your point to introduce RG to study ODEs. So I want to know what aspects of ODE you want to study. For example, what kind of properties of solution you want to study, or you want to study the bifurcation property or stability of solutions. So what kind of problems do you want to study?
So in my case, I want to have the explicit form of the time evolution. So say by using other method, we can have this kind of the form, but it is implicit.
Implicit in what sense?
Implicit in my sense is that we can say the waveform should be distorted or some kind of things, but we don't know how it is how is it distorted, like in the rectangular shape or because by using this method we can have the cosine plus cosine x plus cosine 2x plus cosine 3x, so kind of the form gives the exact form. while it is the approximate solution.
So what's the difference, for example, using classical methods like Fourier transformations or other integration of ODE?
Yeah, well, yeah, if you can solve this ODE by using other methods, please tell me. Yeah, but yeah, yeah, there are many trials to obtain this sort of the ODE with the nonlinearity in biology. But I mean, no one obtained the exact, not explicit time evolution. So this is the first time. Yeah.
Yes,
I'm interested in the relationship between the period of oscillation T and the shape of oscillation LS. So is this, for example, is it similar to the relationship between the oscillation period T and the oscillation amplitude? For example, if the size of the element line is here, the period of element line is here in this case.
Yeah,
But
yeah.
I'm just wondering the relationship between LS and the amplitude.
Yeah, great. Yeah. Great. Roughly proportional. I mean, yeah, so yeah. But we don't know why. Yeah, but yeah, when we do the numerical simulations and test when the waveform is more distorted, so and also the reaction speed increase. When we reaction speed by the increase of the reaction speed when the waveform is distorted, always amplitude is also getting bigger, is accompanied by the more waveform distortion. So that we don't know how it is related. And actually, experiment show that the waveform is distorted and also amplitude becomes larger at higher temperature. So it's an imbalance. Yeah.
so the larger amplitude means larger nonlinearity. So so that is, you said, related to this distortion may correspond to the kind of a non-Gaussian, or where the if it is one, it's just the very beautiful, simple oscillation. But if this NS becomes larger than one, then the wave shape is more distorted.
Yeah.
So you said that these two are related.
Yeah.
Okay.
Yeah. And also it is also related to the phase shift. And waveform is more distorted, it's more difficult to be, the phase is getting more difficult to be shifted. So that if at a higher temperature and like a summer, waveform is more distorted, you may feel more jet lag. I don't try to scale it, but yeah, that is just what our solutions.
順番に、武田さん。あれ私は彼ですね。
I was wondering how both the model and the methods are robust against noise, because I guess I mean, I don't know, maybe if you eat more, then the reactions are not exactly the same speed, or if there is some stochasticity in the equation, can you still apply this method and does it still give you something?
Yeah, I think yes, in the book of the Kurosawa published several years ago, has a chapter of the stochastic models, I guess, but yeah, but I don't know. Yes, eventually, yeah. So yeah, the biochemical noise are often negligible. Yeah, it can happen.
Thank you.
I think that renormalization group and the biological time relation is very interesting. But the renormalization group method is to obtain the better solution for this set of equations. And this set of equations, I think the coefficients remember the 24 hours. And not only self-adjust the period, but the memory may be stored in each coefficients.
Memory.
Memory. I think that not only from this set of equations, you can the temperature dependence introduce the variation of the period. But if you fix the period, then the shape tend to change.
Yeah.
Correct. And then I think this kind of self-regulation is not the only solution for the stability of the biological time, but some memory is set in each coefficient.
Yeah, absolutely right. Yeah, so I have discussed just the oscillation under constant conditions. And with the 24-hour light dark cycle, actually the waveform is changed, and also the amplitude changes. And also, so the related issue can be that if say so the medical doctors or the nurses experience the irregular time schedules, you see, and it can change the modification in circuitry. So so we don't really understand, I think, that modification, but it's a big important problem.
So the only non-linear part appearing in the equation is that F is 3. So can you repeat what X3 is and why that assumption is valid? So what is that X3?
And for X3,
Why? So I have two questions. What that X3 is and why is that valid? The only non-linear part appearing is that.
I mean, yes, f of X3 just like to show that f is it's a function of X3 in a modified protein, and it just try to make a model which can be simpler and it corresponds to the experiments. For the simplicity of the arguments, the decay rate is better to assume the linear reactions. And for the synthesis of protein, and also it should be reasonable to assume that it's linear. And in contrast, the regulation of the gene expression, according to some experiments, it is good, it's okay to, it can be reasonable to assume it is a nonlinear function, the substrate. And yes.
So
this is the f of X3.
I see. So what is that? What is?
X3, f of X3 is the decreasing function of the X3. So actually when we conduct a linear stability analysis, if there is a nonlinearity here, the steady state can be unstable. So with the nonlinearity. Yes.
It is totally a surprise for me that
Really?
someone applied a renormalization group approach to biological time. So you might tell us whether there are simple and natural physics or biology reasons why renormalization group approach might be valid in the biological time. Is it just by accident or there is some natural reason that this was done?
Yes, I think yes, I think there was some answers. Because if we this 24 resistivity is actually the treating the phenomena in a slow time scale. So in that sense, the renormalization group method means, because if I understand correctly, the renormalization group method is good to handle the slow varying solutions. So I think this can be, and the second reason is that because, Atsuha-san is a friend of the Kunihiro-san. So like with answer. Yes, I think the first answer can be better answer. So because this method is good at treating the slowly varying solutions. So
Probably you can solve the set of equations by numerical method.
Yeah.
And that's the most correct one. And you wanted to have the analytic solution.
Yeah.
Right? That's why you needed the renormalization.
Yeah, of course. Yeah. Yes. Yeah. Thank you. Yes.
Maybe somebody, you first ask, yeah.
I have a question. To explain the lifetime period of time starting from the period of this disease.
Yeah.
I think our daily life, our phenomenon, daily life is kind of macroscopic one. So I have a gap between macroscopic life and microscopic origin of the period. So do we have some mechanism?
That's always a mystery of
biology.
biology.
Thank you. Actually, this is a measurement and within the cells. These cells are taken from the human, and if they measure the period of the daily rhythms, the cells, these are stable at the temperature, and the cell division is the transfers at high temperature. And actually, we don't have an answer, but what we know experimentally is that there are the organ with the 100 cells in your brain, and in the mouse brain, and also in your brain. But if you switch the advanced type of mouse, if you switch this organ of the advanced sleep type mouse to the delayed sleep type mouse, to the part of the brain of the delayed sleep type mouse, this delayed one becomes the advanced type. So we know such a relation. And actually, so if the experimental biologists culture these organs in a tube, the oscillation is like this. So I think I don't say that we understand perfectly the connection between the phenomena, the macroscopic scale and the microscopic scale, but we see actually the connections.
Yeah, something to study such a relation. Such a field to study.
Yeah.
So,
actually, no. And if you know the methods, please study biology. Actually, this is the history of biology. I mean, the understanding of the maybe I'm not very present biologists, but so the understanding of the cells and the phenomena within the cells has been done very nicely. But eventually, we want to understand our body as a whole, and we have the gap actually done in the understanding. So, if you have a good oscillating way of understanding, please study biology.
I have two questions. One is how NS is determined by other variables like X, K, or F. That's one question. The other is how your theory can be generalized to other like clocks. Like for example, if you increase the number of interactions or variables, still your theory can still the result is the same.
Thank you for your question. So we have been studying now, and so actually we can do yes, and often this yeah, this P1, P2 can change the waveform distortion. And yes, that has been studied, yeah, numerically. And yeah, it's very important question. And it actually correspond to the what can be observed experimentally. And as for the generality, we have been testing, and yeah, for the moment, by increasing the one variable more, or two variables more, the conclusions the same. But I think that that sometimes the conclusion shouldn't hold. And in that case, we don't know. Thank you.
So my understanding is that you use the renormalization group as a way to find analytical solution for the ODE. So my question is it seems necessary that the ODE should have a oscillatory solution, like periodic solution. It should not be just a single pulse, for example. So if it's just a single function that does not go on and on and on, can you really use this method? Like, is it general to ODEs?
Yeah. I don't know. But so what I know is that this method can be also used for the analysis of chaos.
But that's also oscillatory usually.
Yeah, but yeah.
Is it ever used for non-periodic solutions?
Yeah. Yeah. Actually, I cannot answer. So yeah, what I know is that the limit cycle and chaos.
So
yeah, yes. So yeah, please look at the Kunihiro-san's book from Springer. Yeah, it is. Oh, yeah.
So as far as I know, like geometric technique for the ODE can be applied for the autonomous systems. I mean, that does not depend on the explicitly depend on the time.
Oh, really?
Yeah.
Thank you. Yeah.
Yeah. And my question is there any biological interpretation of the fact that any or the ODEs that describes biological phenomena is autonomous, or that's the most common, or maybe there any reason for the equation for the biological phenomena should be described by autonomous differential equation, the interpretation. My equation of my.
Yeah. What I can say is that I think my answer doesn't really answer to your question, but what we observe is that actually this way of the ordinary differential equations, is sufficient. It seems sufficient for describing the experimental observed phenomena. But as I said, this way of the approach to describe the ordinary differential equation has been challenged. So yes, I think I don't have the answer to your question. Yeah, thank you very much. Of course, the noise also is important. Yeah. Yes.
So back to a naive question. How can a baby acquire this skill?
Baby? Yeah.
How can a baby finally acquire this?
Yeah. Thank you. Yeah. Actually,
Baby sleeping or
But it seems random for
Actually, I think it's not. And I see, actually, according to the experiments in mouse, before babies are born, actually circadian rhythms is generated. If you take if you take some cells after fertilizations, and at some time point, the oscillations can be autonomous oscillation with a period of 24 hours is generated. Actually, in contrast, the sleep of the babies doesn't show really the 24-hour rhythmicity, and we don't know why. But we think, and I naively think that it can be because of the two factors. The one factor is that desynchronizations of the oscillations. The oscillations can occur at the cellular level in babies, but because the synchronizations, the coupling between oscillators are weak, they cannot show the 24-hour clear oscillations. And second reason can be they need to eat every 2 hours or 3 hours because their digestion ability is limited. So yeah, so yes, I mean we don't know here how the baby sleep is generated. Yeah.
Yes.
Yeah, no, just another question. I read a book about circadian rhythm recently.
Oh,
Yeah, and yeah, I wonder that, and I heard that the circadian rhythm changes as we get older. Right?
Good.
Yeah, and that kind of phenomenon can be described by the.
Yeah. So yeah. My answer should be yes, but yeah. But naively, yes, experimental it was observed that as you get older, the amplitude of oscillation becomes smaller, and wave distortion seems changed also. And so yeah, mitigated also the change in jet lag effect by aging. This is still overall. Yeah.
Any questions more?